nível genômico amplo. José Sotelo Silveira é professor nas áreas de genômica e biologia celular e molecular, com atuação no Instituto de Investigaciones Biológicas Clemente Estable e na Facultad de Ciencias da Universidad da República, em Montevidéu, Uruguai. Sua pesquisa foca em áreas interdisciplinares da biologia molecular, genômica, buscando avanços no entendimento dos mecanismos biológicos fundamentais. Aí passo a palavra para o nosso coordenador do CDTS, Carlos Morel, para dar as boas-vindas ao nosso convidado.
Bom dia a todos. Damos início aqui a mais um seminário CDTS, hoje com o prazer de um convidado do exterior, nosso colega José Sotelo, que eu tive o prazer de conhecer durante o evento em Caxambu, os 50 anos da pesquisa básica em chagas, e vi que ali o trabalho dele tem tudo a ver com a Fiocruz. Então, aproveitando, convidamos ele passar aqui uma semana para um contato mais pessoal. Então, um grande prazer estar com ele aqui e espero que, ainda nesses dias que ainda temos aqui e mais para frente, que surjam oportunidades de colaboração entre a Fiocruz e o Instituto de Investigações Biológicas Clemente Estable. José.
Muito obrigado pela invitação. Está fazendo um tempo muito lindo aqui. Gostei muito de conhecer vocês. E now I will switch to English because my Portuguese is really bad. So let's see if I could put. Okay. So the main area of my work is related to translation and basically trying to understand translation at the genome-wide level. At some point in my career, due to the number of people working in Trypanosoma Cruzi in Uruguay, I started to really pay attention to this beautiful model. And I went from cell biology and neurosciences into trypanosome biology like probably 15 years ago, and started to work in collaboration at my institute, which is named after a school teacher that was self-taught and worked with the Nobel Prize Ramon y Cajal in the beginning of the 20th century. And on the way back, started a research-only institute in 1927. Now, a few years from now will be our 100-year anniversary. So it's a very old research institution in Uruguay. I run the Department of Genomics at my institute, which also ran a small genomics platform. And with that, I will go into the main subject of the talk, which is trying to understand translation at the genome-wide level in Trypanosoma Cruzi. The first slide, I think you have to touch the screen.
Pessoal, desculpa interromper. A tela não está compartilhada com a gente.
Okay. I will figure it out. I think it's like this.
Go to the other Zoom window. Yeah. What I'm about to say. What is?
Are you seeing the?
Okay.
This is the
Yes.
full screen.
Perfect. It is
Okay.
perfect. Thank you.
No, thank you that you warned us.
Agora a gente pode baixar aqui.
Podemos baixar isso aqui.
Ali menos aqui o sinal esse primeiro. Isso.
Aí. Perfeito.
Isso.
Então, esse é o símbolo do meu instituto. Voltei para o português e não é muito bom. So, going back to English, this is my institute and this is my department that I run, and I was starting to, with acknowledgement to the group, this is a 2 or 3 year old photograph. Martin, Santiago, Rafael, Pablo, and Juan are the main guys involved in this, in the work I will show you, and also done in collaboration with Professor Garat and Professor Duhagon in the School of Science at the functional genomic department, and with Thomas Kieslinger at the University of Toronto in a proteomics lab. So the talk will be divided in 3 parts. The first one is how to study translation at the genome-wide level. Sorry for the Spanish word here, right? And then go for the main results that we have published in the area. And then I will share some unpublished results about the translation machinery, protein composition, and associated non-coding RNAs, the rank RNAs in the translation and machinery of Trypanosoma Cruzi. Everything will be in Trypanosoma Cruzi. So how do we study translation at the genome-wide level? In 2009, a nice paper by Nicolas Ingoglia opened a very wide door to understand or measure translation at the genome-wide level. By that time, I was doing my postdoc, and I was trying to understand translation using microarrays, which was not, at this level. And I switched to this technology, which I will explain in a few slides forward. Basically, ribosome profiling centered technologies to track the position and number of ribosomes on mRNAs being translated at a given moment in the cell, in your experiment on the cell biology of what you are studying. And basically, you do a nuclease treatment like an RNase protection assay, protecting the ribosome, the fragments under the translating ribosomes, and then you prepare the library, you deep sequence that, and then you do some bioinformatics to understand where those ribosomes were located. And you compare and normalize on the baseline by simple RNA-seq procedures to understand the relationship of the levels of mRNA to the levels of ribosomes on top of those mRNAs. So meaning that you will have a clear idea of at least how many ribosomes you have on translating ribosomes, on translating mRNAs. So it's a technique that gives you single nucleotide resolution at the genome-wide level. Basically, you can measure translation on all the mRNAs that can be detected in an experiment. Basically, you go and install the translation using cycloheximide or other drugs that can stop translation at different points, like initiation, or you do rapid freeze. There are many options. You lyse, and then you do the RNase protection assay. And then on the same population of cells, you also extract the mRNA and do regular mRNA-seq. And then what you get is a picture of exemplified that with this figure. If you have many ribosomes at the same position, when you map the footprints onto an mRNA, you will have a peak here, but then you have distributed signal along the mRNA, and the whole amount of footprints in that message implies, or is a proxy of translation of that message. This allows precise quantification of the proportion of mRNA actually bound to polysomes and allows the study of ribosome dynamics during translation. A number that is used frequently is the translation efficiency. So it's basically a ratio between translation level derived from the footprints, right, of the ribosomes compared to the steady-state levels of mRNA levels coming from RNA-seq. So you can have movements if you compare proteins like activity of synthesis against RNA abundance. Sometimes you get an increase in mRNA abundance together with an increase in translation. So you have a diagonal move in this example. But you could also have situations where the mRNA doesn't really change, but the level of translation and the number of footprints change in the vertical line. Right, so you will get a precise vision of something happened after transcription, before translation, something in that compartment, something in that translation compartment, right? From that, you can do many things, but it's clear that the correlations levels with the transcriptome, to the translatome, to the proteome vary in different numbers, being closer between the transcriptome and the translatum, or the translatum to the proteome, but very far from the transcriptome to the proteome. Thing that is known, but is clearly observable or seen in this with this methodology. As another way of looking, this is a very basic example, a HeLa in control plus some growth factor. When you evaluate the translation as a fold change in the changes between these two conditions, you could have translation only changes when no transcriptome is changing, or homodirectional changes where you have increasing mRNA and increasing translation seen in green here. But all the quadrants here are important when you study a phenomena of regulation. So the technique is quite powerful, not only to do genomics. It's a cell biology tool. That's what I like about this technique, because you can really ask about the cell biology of translation regulation. Once you have, for example, these yellow dots here or these yellow dots here, you know that some particular regulation is happening at the level of translation, not at the level of transcription. And if you add proteomic information, you may also figure it out that is at the level of translation, not impacting in the final state of the proteome, or also impacting in both, right? So you could do nice correlation when you do multi-omics to figure it out, regulation steps. For sure, translation can be regulated at many steps, at the initiation, elongation, and termination. And if you increase the complexity of the experiment or the variables you are trying, you will get interesting views of what's happening in translation. I mean, no doubt translation regulation is a major contributor of gene expression level, right? And but for example, to exemplify, it's important for rapid responses. For example, you don't need to go through transcription. You can jumpstart translation of some mRNAs, just as key examples to understand the power of the technique. So, going into the second part of the talk, I will tell you about our experience doing some research or doing some investigations on how important is translation in regulation in T. Cruzi. As you all know, if you think in T. Cruzi, you have almost no regulation at the level of transcription, it's polycistronic, and you have proposals, or well, it's already known that you do regulate at the steady state level of mRNAs, that level through RNA binding proteins, regulating stability, for example, but also after our work and other people working in Trypanosoma Cruzi, it was clear that at the level of translation, you have a lot of regulation too. So we have been investigating transitions in the life cycle and also cell cycle transition in Trypanosoma Cruzi. Here's basically what we do. We compare epimastigotes to metacyclic tripomastigotes, and also G1 to S transition, right? Using riboseq and also proteomics. Now that you have seen what I call the dots of, the clouds of dots, basically, you can see an overall picture of a comparison of the fold change between a metacyclic stage compared against an epimastigote stage at the translatum and at the transcriptome. Basically, we see mainly a diagonal here, implying that you have altogether changes in transcription, but also changes in the translation between these two stages. The green dots here and the red ones are mainly mRNAs that are being regulated at the level of translation, And interestingly, the blue ones and the yellow ones are mainly at the level of steady-state regulation of the stability of the mRNA. And this is when you have a very huge differentiation transition. But when you are in the epimastigote, where you have a phase of the cell cycle being studied, the cloud of dots is quite different. But it's interesting that the main change is seen in translation. So when you have a rapid phase transition, which is ours, right? These are cytometry synchronized and purified populations. When you have that transition, you mainly see changes at the ribosome profiling. Sorry, this is an R, meaning this is the translatome. It's exactly the same axis as this one. But you don't see the transcriptome changing much. The orange dots here are not spreading on the x-axis. So we are going to go a little bit into detail into these two models. And you will see, when you see this sign here, we will be talking about this differentiation transcription. And if you see the cell cycle, is that we are talking about the G1 to S transition. Interesting aspects. This is now an old result, but I'm fond of showing it, because when you detect mRNAs by RNA-seq in the epimastigote and the metacyclic parasites, you basically detect almost everything in the two stages. This is detection. I mean, it means expression. I'm not talking about regulation, but if you go for a scatter plot for the RNA-seq in one stage and the RNA-seq in the other, this is actually one of the first transcripts of showing steady-state regulation. mRNA steady-state regulation in Trypanosoma Cruzi. We did it in 2013 or something. And you see, these changes here, these dots marked in orange or red, are changes in mRNA stability. When we look at the translatome here down in the mark in green, are the genes detected being translated in common from the epimastigote to the metacyclic. Interestingly enough, there are mRNAs, about 2,000 mRNAs, that are not translated at all in the metacyclic. This was, of course, already known, because the metacyclics are quiescent, and they are really low metabolism, waiting for infection. But this gray crescent here implies that they are not loaded in the ribosomes. But also, you have this other one here that is only specific to the metacyclic. So you load some stuff in the metacyclic, and of course you maintain some stuff in the ribosomes. But these 5,000 mRNAs are spread across this scatter plot, and you see a lot of regulation here. And here you are comparing the translation in the metacyclics versus translation in the epimastigote. The heat maps are on the right side, just to see it in another way. So different groups of mRNA go up and down, and they can be clustered. Another thing interesting about this technique is that you can try to quantify the level of change, the magnitude of the change. Because if you see the fold change, I mean, this is a box plot indicating the main difference, for example, of fold change between the two condition studies. The ribosome footprints or the translational footprints have more spread than the RNA-seq stuff. So it means that there is more regulation in translation than in transcription. And here the mRNA is downregulated and upregulated as separated. The upregulated even have more spreading in the footprints, right? So the magnitude of regulation in Trypanosoma Cruzi is quite large in comparison of the regulation of the steady state level of the mRNAs. Another thing you can do, and we can go on forever on these type of things, is which proteins are specifically being regulated. So we went after, of course, the most famous two. If you go to an infected state, transialidases should go up, at least the most important one in infection, and that was true. Remember, transialidase is a huge family, quite similar among them, and are all marked here in red. And you see they have a diagonal component, but not more close to the vertical than to the diagonal, right? So it means that it's more translatively regulated as regulated at translation and a little bit at transcription. And if you measure the efficiency, remember that parameter that correlates how many ribosomes you have on those mRNAs against the total population of mRNAs, it's clear that these transialidases are very lowly translated in the epimastigotes, and they go in increase in the metacyclic stage. The other classical example at that moment was the one that we needed to do is the ribosomal RNA. Sorry, the ribosomal proteins. Ribosomal proteins are highly transcribed in the epimastigotes, right? And highly translated in the epimastigotes, compared to whatever is transcribed, all the mRNAs being transcribed or all the mRNAs being translated. So they are highly translated. You can see the comparison here for the ribosomal proteins here, right? But what happens, this is the epimastigote stage. What happens if we go to the metacyclic stage? You get a decrease here. This is the mRNA level at the metacyclic, the mRNA level, and this is the translation level of the ribosomal proteins. Goes from here to here or from here to here. So you're repressing translation of the translation machinery. So that was known too, because translation rates go down when you go to the infective side. But this is the demonstration that the regulation is happening at the level of translation, because you have mRNA, you have a lot of mRNA, but you don't translate that mRNA. When you go for the cell cycle, I have several things to show, but let's pay attention to these clouds here. This is the cloud I show you, where you compare the ribosome footprints or the ribosome profiling to the transcriptome, and you see more on the x-axis the movements of the translation. If you have a proteome data set that we did for this experiment, you could also see that the main change is also in the proteome, right? The transcription doesn't change, and the proteome also changes in the same direction as the translation, right? But if you compare the proteome against the ribosome footprints, meaning whatever is being detected at the steady state level of the proteome, against whatever is being translated, the cloud changes and you have more quadrants to pay attention. And here are the percentage. For example, 30% of the mRNAs, when you do this comparison, are changing only in the ribosome profiling. Here is also a similar percentage when you go from proteome to transcriptome. And then you have a more spread variation when you compare the proteome to the translatome or the ribosome footprints. Sorry if I changed the words, but it's interesting to see that all the quadrants can be quantified when you add another omic layer of information. And this is just to show you an idea that we published, trying to cluster different groups of genes that have, for example, we have here like 8 clusters where translation, marked as R here, ribosome footprints, behave differently along the clusters, and the proteome behave differently. And to make a long story short, analyzing the quadrants correlated only by the behavior of expression, we ended up analyzing different set of spots. This cluster correspond to an inhibition on transcription and translation, sorry, on proteome, but you could go over this watch, and the conclusion that we have, we need to do more experiments, but we see is that when you are studying G1, you repress stuff that you need for S, and then when you go to S or G2, you start to express whatever is needed from S or G2, and there is some asynchronous behavior between the components. We need to do more experiments, but basically we got a hint of what was happening at different stages of the cell cycle. You don't have, sometimes you do have coordinated increase in translation and translation and proteome, but sometimes you have some minor uncoupling. For example, I think cluster 3, it's something that we are seeing on S. So you are increasing cytoskeleton and mitosis machinery a little bit earlier than when mitosis happened, something like that. So this is just one thing that you can do when you're analyzing fine stuff. We have also data from S to G2, which also confirms a little bit this idea. So now I'm going to share with you some unpublished results. So Felipe, if you can stop the recording.
Ok, estou parando aqui.
Perfect,
Okay.
perfect. So when I was carefully studying these